name's Sammie. I'm working on an Oscar project this summer with the Potomac Environmental Research and Education Center, and I'm creating a food web with the fish community in 2 Potomac River tributaries. I'm hoping my project will be able to tell us what our fish are eating in the ecosystems, and that way we will be able to trace where pollutants and micropollutants could possibly transfer as it moves up the food chain. So in order to get my samples, we go to 2 different locations. We collect our fish using 3 different types of nets in order to sample different habitat types within the 2 tributaries. And then once we have our fish collected, we take them back to the lab and we remove their stomachs and then go through the contents under a dissecting scope and count the number of organisms and assess the percent of each in the stomachs. An interesting part of my project is looking at the difference in fish communities between submerged aquatic vegetation habitats, so places where there are plants growing underwater, and then non-vegetated habitats, so where there aren't as many plants. In Gunston Cove specifically, it used to be algae dominated, where there would be a thick green layer of algae on the water and the submerged aquatic vegetation couldn't grow. We've now cleaned up our water so there aren't as many nutrients in it helping the algae grow. We're shifting to more SAV dominated system. I really like the team aspect of this Oscar project because it helps me understand how my individual project is tying into a bigger picture. I'm creating a food web for the fish communities to see what they're eating within each system, whereas the chemistry team is assessing the micropollutants within the locations. Our projects combined will help us better understand ways that the micropollutants could travel and possibly impact human consumers, such as people maybe fishing recreationally at the locations.